Hello. In this session, we're going to talk about Chapter 22, which deals with our immune system. In this particular section or session, we're going to talk about some infectious agents. So what are things that can cause infection to our body? And then we'll talk about the innate immunity. And then in the second section tomorrow, we're going to be talking about adaptive immunity and how the two relate with each other. So in chapter 21, we covered the lymphatic system, and we're kind of going to tie that into chapter 22 because those two are very closely related to each other. Before we begin with the immune system, let's talk about what are some things that can cause infection to our body. So what are some infectious agents that can serve as pathogens to our body, which is pathogen is something that causes harm to us. So probably the most commonly known pathogens would be bacteria and viruses. Bacteria are single cell prokaryotes. Prokaryotes mean that the cell doesn't have a nucleus, and the DNA is just freely in the cytoplasm of the organism. The bacteria are living things. They can be up to 10 times smaller than our body cells, so they're pretty small compared to our cells. Often bacteria have capsules. These capsules or the capsule of the bacteria sometimes helps with the bacteria being more virulent or being more capable of causing disease. Just a little harder for our body to recognize them because of this capsule. And bacteria often release enzymes or toxins, and that is really how they cause harm. So it's the toxins that the bacteria in our body releases or enzymes that are going to harm our cells. And that's bacteria. Viruses, on the other hand, are not alive. So as we said, bacteria are living things, they're cells. Viruses are not cells. Viruses are composed of a protein coat. So you have a protein coat, just some kind of a shell, and then inside of that protein shell, you can find the genetic material. And this genetic material could be either DNA or RNA. If you've ever heard the term retrovirus, retrovirus is a virus that has RNA as their genetic material instead of DNA. And so coronavirus is a virus. So when you see the picture of it and what it looks like, it's round and it has those proteins sticking out of that. We call viruses obligate intracellular parasites because viruses are not able to reproduce on their own. So obligate means that they have to do something. Intracellular means that they have to enter the cell, and then parasite is something that harms its host. So the way that the viruses cause harm to our cells is that they will either the whole thing will enter the cell, and then the DNA from that virus will merge with the host cell's DNA, or some viruses will inject DNA into the host cell, and then that DNA will merge with the host cell's DNA, and it reprograms that cell and ultimately causes cell death. It causes the host cell to create more copies of the virus, and then eventually that cell dies. So that's viruses, and they're not, as I said, they're not alive. Sometimes this is when people are thinking like, okay, well, what do you mean they're not alive? What do they mean when they say that you're getting a vaccine that contains a live virus? And what that means is that that virus has not been broken into pieces, but it's been weakened another way. Okay. But they're not really considered living organisms. There's no cell membrane, no organelles. It's just a protein with some DNA or RNA. All right, so that's viruses. Next group are fungi. Fungi are eukaryotic cells, so these do have nucleus. The single cellular fungus is yeast, and then we have a lot of other multicellular fungi like athlete's foot, ringworm. All these are fungal infections. Protozoans, next category. Protozoans are unicellular eukaryotic cells, and these include like amoebas or some other organisms. The organism that causes malaria is also a protozoan. And then we have multicellular parasites. And multicellular parasites, this is going to be the worms. Okay, so there's a lot of different kinds of worms that could infect human body. For example, there are some filial worms or flukes, different kinds of flukes. There's tapeworm, which would be in your digestive system. So a lot of different worms can serve as parasites and cause harm to our body. And then so these infectious agents are summarized in your textbook in table 22.1. I think it's 22. Yeah, 22.1. And there's one thing that's missing though in this table, but it is mentioned in the text, and that is prions. Prions are infectious proteins that we don't really know where they originated from, but they're proteins that can cause harm. And in animals, probably the most well-known prion disease is mad cow disease. Okay, this affects cows. But there is a human variant of this disease, and that's Creutzfeldt-Jakob's disease. And this basically attacks, I'm not going to go into too much details about this, but it basically destroys your brain. And once a person contracts it, they would be dead within about a year is the life expectancy of death. But we don't understand a lot of stuff about prions. First of all, where did they come from? We don't know. So there's a lot of research that's going on with prions, and there are other diseases that are also caused by prions, not just this one. That's just kind of a most well-known one. And so that's our infectious agents. Let's summarize a little bit our immune cells that we've already talked about immune cells in chapter 18. So you were supposed to look at different types of immune cells and just kind of their general function. I just want to remind you, so this is not going to be new stuff, but it's kind of just a reminder. We have two main groups of leukocytes. We have granulocytes and agranulocytes. Granulocytes are the phils. Okay, granulocytes are the neutrophils, eosinophils, basophils. Agranulocytes are the cells that do not have granules in their cytoplasm, and these include your monocytes, which will become macrophages, and then B lymphocytes, T lymphocytes, and also natural killer cells, so all the lymphocytes as well. We are going to look at their specific function quite a bit in this chapter, in chapter 22, so I'm not going to get into that at this beginning of this section. As far as location of immune cells goes, so where are these immune cells? We know some of those immune cells are in our blood, but our blood it's not a huge percentage of our blood that contains leukocytes, right? It's about 1% or less of our whole blood that is composed of white blood cells. And that's because our leukocytes are found in other places. Okay, our immune cells, they are mostly found in other places. So lymphatic tissue, remember those secondary lymphatic structures that we talked about in chapter 21? Well, that's where your immune cells are. We've mentioned dendritic cells, we've mentioned lymphocytes, we've mentioned macrophages. Okay, so that's secondary lymphatic structures are designed to house these immune cells. We can also find various immune cells in different organs. So, for example, we'll see alveolar macrophages in the alveoli of the lungs. Okay, and then sometimes these immune cells, when they're in a specific organ, they have a specific name, like alveolar macrophage is in the alveolus of the lung, and so that's why it's called alveolar macrophage. We'll see Kupffer cells in the liver, and these are going to be special types of macrophages as well, and we'll talk about those in digestive system later. And yeah, so that's organs. We can also find immune cells in the epithelial layers of the skin and mucosal membranes. And we see dendritic cells, a lot of dendritic cells here. And I think there is a picture right here. So let's see if we can kind of get a visual of what a dendritic cell looks like. Dendritic cell is named dendritic because it has dendrites. Okay, so it has these projections. So look at this. Okay, skin and mucosal membranes. You know, the cells of the skin and mucosal membranes are really tightly packed. And so this dendritic cell, which is this yellow one right here, those dendrites can squeeze into spaces between the cells. And so the dendritic cell kind of has like a longer reach to monitor like a bigger area around its body. Okay, so that's our dendritic cells. And then connective tissue is underneath this. And connective tissue is going to have two main types of cells, mast cells, which are very similar to basophils. They release the same chemicals, and they really complement basophils here. So they release histamine and heparin. Histamine is a vasodilator. Heparin is a substance that prevents blood clotting, so it ensures that blood flow is continuous throughout the area. And yeah, you can find these mast cells and basophils in mucosal linings, in dermis of the skin, also connective tissue of various organs and such. So different areas prefer different types of immune cells, but you have immune cells pretty much everywhere, almost everywhere. There's one thing that the immune cells kind of have in common. So all the immune cells are going to have a different function. They're going to work a little differently. But one thing that they have in common is that they release cytokines. And cytokines are soluble proteins. These soluble proteins are produced by these immune system cells. So I will remind you, like when we talk about a certain protein, I will be like, okay, this is a cytokine. Okay, so cytokines are soluble proteins produced by immune system cells. And they can have various functions. They regulate and facilitate immune response in your body. They can serve as messengers or messaging molecules. They can regulate inflammatory response. Sometimes some cytokines are actually used as weapons. So when we look at a natural killer cell, it's going to use a cytokine as a weapon. And cytokines also influence other cells, so non-immune cells. Especially when we're talking about the function as a messenger, okay? Immune system cell or immune cell can release the cytokine, and then it can serve as a message for a non-immune cell. Both the interferon is a type of cytokine, and that can serve as kind of a warning mechanism for a healthy cell. The action of cytokines is similar to hormones. So there's a receptor for the cytokine, and then there's a action that will happen following the cytokine binding to the receptor. Cytokines don't have a long half-life, so they have a very short half-life, and that's good. You want to release them, send a message, or do whatever they need to do, and then they're gone. They're degraded. Some examples are going to be interleukins. We'll talk about interleukin 2 and 4 specifically later on in the adaptive immunity. And then tumor necrosis factor, which its main function is to kill tumor cells. Colony stimulating factors. So if you recall from chapter 18 when we talked about hemopoiesis, there was a bunch of colony stimulating factors. Those were things that were deciding which way the cell was going to develop, and as well as interferon. I've already mentioned interferons. So that's kind of just like an overview of different cells. We're now going to jump into innate immunity, and I've already mentioned that that term, innate immunity. So just kind of as an overview, like an introduction into the immune system. Okay, this is a summary of what we're going to be talking about. All right, this is how your immune system is divided. So we have innate immunity, or innate immune system. This is also known as the non-specific immune system. This is something that you were born with. It's something that doesn't really need to develop. You already have it. And then on the other side, we have adaptive immune system. And this one is going to be designed specifically against that pathogen, whatever that pathogen is. And it will always follow innate immune system. So we start with innate immune system first, and then adaptive immune system will be activated later. Innate immune system is actually like if we talk about lines of defense, because your textbook kind of talks about lines of defense. So there's a first line of defense, second line of defense, and third line of defense. The very first line of defense that your body has is your skin and your mucosal membranes. This is what is designed to prevent entry of pathogen into your body. Okay, so we say right here that this is first line defense. Okay, this skin and mucosal membranes. Okay, so skin is a barrier from pathogen or for pathogens. They cannot burrow usually through a skin that's not been damaged. Okay, so if your skin is healthy and not punctured, then it prevents things from coming into your body. Mucosal membranes contain mucus, and this mucus is going to trap any bacteria, viruses, pathogens before they can actually start causing harm. So the mucosal membranes are a form of protection. All right, so that's our first line of defense. Now imagine that the first line of defense, it's like a wall, okay? Imagine that this first line of defense fails, and your wall falls, and somehow there is an influx of pathogens into your body. Then what your body is going to do is it's going to initiate second line of defense, okay? And that's this non-specific internal defenses. The keyword here is non-specific. This is still non-specific. It works against any type of bacteria or any type of virus. Now, granted, some immune cells prefer to fight bacteria, some prefer to fight viruses, some prefer to kill things. But what the key point here is that it doesn't matter if you are, a flu virus or whatever virus. Okay, it's going to work against that virus. Excuse me. Yes. Thank you for my present. I love you. Close my door, please. I'm sorry. I got a present for my daughter. All right, so that's our second line of defense, okay? Non-specific, it works against bacteria or virus or whatever it is, kind of in the same way. And then after as those non-specific internal defenses are happening, those non-specific internal defenses are actually going to initiate the third line of defense, which would be right here. Third line of defense is our adaptive immunity. And we don't, that's it. Okay, so we have first line of defense, our skin and mucosal membranes, that's preventatory. Then we'll have some internal defenses that are going to be non-specific. It's not going to matter, what kind of virus or bacteria or pathogen you are. It's just going to be non-specific, but it's going to happen internally. And then the third line of defense is going to be this adaptive immunity. I'm having horrible allergies, so I apologize if I sound a little not that great. All right, so we've covered the skin and mucosal membrane and how that works. Let's look at the non-specific internal defenses here. More specifically, let's look at these cells here now, okay? So I'm going to be talking about this part right here, and that's cells. And actually, there's another picture here that shows us, who does what. And so here we're going to focus on different types of cells and how they fight a pathogen. All right, so this first group of cells are cells that like to engulf things and destroy them internally. So we have a dendritic cell, or this could be a macrophage, as well as neutrophil are going to be your engulfers. Okay, they're phagocytes. They are eating things. Okay, so they literally will wrap their membrane around the pathogen and then create a vesicle of that pathogen, and that will fuse with the lysosome. The lysosome has digestive enzymes. It will literally cause the pathogen to dissolve, to fall apart. Whatever is left over is then eliminated by the cell into the interstitial fluid. Okay, wherever the cell is, it's going to eliminate the residue. Now, remember those lymphatic capillaries and how they absorb some of the interstitial fluid? So there's an infection, and this macrophage, or this phagocytic cell, engulfed and destroyed the pathogen. The residue or fragments of that destroyed pathogen now are part of the interstitial fluid. So when a lymphatic capillary takes fluid from that area, that fluid is going to contain traces of the pathogen, and that will activate your lymph nodes, and then you have your adaptive immune system, your third line of defense, being initiated. So that's our engulfing cells. We have some cells that are going to serve in the pro-inflammatory response or inflammation response. So there are pro-inflammatory secreting cells, basophils and mast cells. Okay, they release histamine. And again, histamine is a vasodilator. So right now in my body, somebody is releasing a bunch of histamine because I'm going through this allergic reaction. And that's why, you know, all this kind of fluid is leaky right now because histamine is a vasodilator. So it opens up your vessels. It increases blood flow to the area and also increases capillary permeability. And so that's why that fluid exits from your cardiovascular system. Another chemical that is being released by basophils and mast cells is heparin. And heparin is an anticoagulant, so this prevents blood clotting. So this ensures that this area has blood flow through this area continuously, okay? And then there's some eicosanoids. Eicosanoids are those local messaging molecules, if you remember from chapter 17. And they just, in general, increase inflammation. We are not going to really talk about specifics of that. Histamine and heparin are the two big ones right here. And then we have natural killer cells. Natural killer cells are designed to kill infected cells. And we'll see how natural killer cells could be activated later. But if a natural killer cell detects that your body cell has been infected, this infected cell needs to be destroyed. Your body doesn't heal cells. Okay, we can make more cells. If the cell is sick, it needs to go. It needs to be destroyed. And so what the natural killer cell is going to do is it's going to release perforin and granzyme. These are cytokines, both of them. And this is an example of how cytokines can serve as weapons. Perforin, if you think about the word perforin, you know the word perforate or if something is perforated, it has holes in it, right? And so perforin is a protein that will basically attack the infected cell's cell membrane, and it will create a hole in that cell membrane. And then this granzyme is an enzyme that will enter through that opening inside of the cell, and it will cause the cell to go through apoptosis. Apoptosis is a cell death that makes the cell look kind of like this. It makes the cell kind of shrivel up on itself and die. That way, you're containing that infection in that shriveled up cell. And it's now smaller than the original cell. And you can have macrophage or another phagocytosis performing cell come and engulf it and destroy it. Okay, so that's what our natural killer cells do. And then the last group of cells that we'll talk about are eosinophils. Eosinophils are cells that are designed to really be effective while fighting multicellular parasites. So when you have like a parasitic infection, like a worm infection, something like that, your eosinophils would be the ones that are getting activated, that are actively fighting that worm, okay, or that parasite. So that is the cellular defense. Now, let's go back to our diagram. Okay, remember this diagram right here. And my markings went away at this point. But we just talked about, so we talked about our preventing entry. We now talked about the cells and how the cells specifically do what they do. Notice that this was all non-specific, okay? So like the neutrophils and dendritic cells, okay, those cells, the engulfing cells, they didn't care what kind of bacteria that was. They knew it was bacteria and they engulfed it, right? Or your eosinophils are designed or are working against parasitic infections or parasites, multicellular parasites. They don't care what kind of worm it is, okay? If it's an eosinophil, it's going to attack that worm or the multicellular parasite. Okay, so those were internal defenses as far as cellular response goes. Okay, we're now going to look at some chemical processes that happen, and then lastly we'll look at physiological responses. Okay, but we're still talking about non-specific events here. Okay, so let's look at the chemicals. There are several chemicals that are involved in the non-specific internal defenses. For example, our complement system. And complement system, let me see if I have it here. Complement system is kind of summarized in this table right here. Complement system is a system of proteins or complex of proteins, and these proteins are they can do several things. Okay, so one thing that these complement proteins can do is they can attach to the pathogen, like right here. So you see that C right here, that's the complement protein. So it can attach to a bacteria or a virus, and it can cause opsonization. This word opsonization, we'll see it again in adaptive immunity, but opsonization basically means that you're marking something for destruction. When that complement binds to that bacteria, the macrophage is going to be more likely to detect it. It's more likely to be seen by the macrophage if the complement is there. So it's like a little flashlight or a little tag that makes the pathogen more visible to our immune cells. Okay, that's opsonization. Complement system just in general helps with inflammation or inflammatory response. So it helps with attracting various cells as well as it does other things. So just supports inflammation. You don't need to go into detail about how it supports inflammation at all. So that's that's the inflammation. It can also cause cytolysis, and cytolysis, cyto for cell, lysis for falling apart. So this complement system can arrange itself on the surface of a bacterium and form a MAC protein. That stands for membrane attack complex. And when this protein assembles itself on the cell membrane, it causes the cell membrane to fall apart. That cell membrane no longer holds together, and the pathogen dies. And then the last thing that the complement system helps out with, and this is pretty important, is that it helps eliminate antibody-antigen complexes. So far, we've only talked about antibody and antigen with the blood typing. But what I want you to know is that the antigen can be anything that's foreign to our body also. Okay, so antibodies are designed against specific antigens. In blood typing, we just talked about antigens A's and B's and the Rh. But whenever you get exposed to some kind of an infection, what your body does is once it recognizes that infection, that bacterium or virus or whatever it is, it will design specific antibodies against that particular antigen, okay? And then when those antibodies are formed, they will then attach to that antigen. So this antigen right here, okay, imagine that this could be a bacteria or a virus, or it could be wrong blood antigen too. Okay, well, I don't know where that went, but that's what I typed. I guess it went off a little bit. I don't think I can. Nope, I can't pull it. Okay, so the antigen could be bacteria, virus, wrong blood type. Okay, that could be an antigen also. What the complement does is it attaches to the antibody at the stem region right here. And then when it attaches to that area, it serves as a link between the antibody and the erythrocyte. This is not agglutination like we saw in the blood typing, okay? Agglutination was when the antigen and the body were joining together. But when complement links the antibody to the erythrocyte, the only reason why this happens is so that the erythrocyte can serve as a tow truck for this antigen and the antibody complex and take it to the liver so we can destroy it, okay? It is what happens after agglutination happens, all right? It is what happens after antibody and antigen interact with each other, okay? So don't get confused, the complement linking the antibody to the erythrocyte with the reaction during blood typing, okay? It's not the same thing. In the elimination of immune complexes, the complement just serves as like a link between erythrocyte and antibody so that erythrocyte can pull it to liver or towards liver. So that's complement system. And that's all you need to know about complement system. The other chemical that's important are interferons. And let me find interferon here. Okay, this is a diagram of action of interferon. And at first, when you look at this, it might look very confusing, like I don't know what's going on here. Well, I know what's going on here, but don't get scared of this and the abbreviations. You don't need to go into the alpha, beta, and gamma interferons. Just be aware that there are different types of interferons. But in general, interferon does two main things, two important things in our body. Interferon is, first of all, it is released by an infected cell. So if you got infected by a virus, okay, you're a cell and virus entered you and you're now doomed to die. You know you're going to die. And that virus is trying to reprogram you. If you're that infected cell, you will release this interferon before you go completely nuts and start making new copies of the virus. You will start releasing this interferon. And interferon does two things. It goes to normal healthy cells, right here, and it tells that healthy cell, hey, there is a virus going on, protect yourself, okay? And so what that healthy cell is going to do is it is going to create or synthesize these enzymes inside, and those enzymes will prevent the virus from replicating itself inside of that cell. So it is, even if that virus would enter that healthy cell, if it received the message from interferon, that cell is protected and is able to basically ignore that virus. The other thing that the interferon does is it is going to send a message out there to the immune cells that I have a capability of killing cells, which would be either your natural killer cell or a macrophage, whoever is closest. Okay, whoever receives the message first and is the closest and can kill the cell fastest is going to be the one that ends up killing that cell. So it sends a message to the immune system cells, I'm infected, I need to be destroyed, come and hurry and destroy me, okay? So you can have, like, if the natural killer cell kills this cell, then after apoptosis, then the macrophage would eat it. Okay, or it could just go into the macrophage. So it could go either route there. And so that's interferon. And going back to our table here, so we've covered our cells. Okay, well, we did this. We cover chemicals now. And now we're going to look at the last two things of innate immunity, and that's the physiologic responses, and that's going to involve inflammation and fever. The inflammation process is going to be a non-specific response in vascularized tissue. Okay, and I'm going to show you a picture that we use here to describe inflammation. Okay, so again, inflammation is non-specific response in vascularized tissue, right? So we have a picture here of a kid that fell off a skateboard and scraped their knee or their skin. Now your first line of defense has been compromised. Your skin is broken. And this allows bacteria to enter, right? So the bacteria is entering into our tissue, and they are looking forward to start infecting you, right? So what is happening following an injured or a tissue injury is the injured tissue is going to release chemicals. Also, these new bacteria or these new pathogens, these cells that are normally not in your body, are producing chemicals that are not native to our body, not normal to our body. And those chemicals are going to activate some of our immune cells. More specifically, your mast cells are pretty important because mast cells are found in your connective tissue, right? And so mast cells are going to detect this. They're going to detect the damage and presence of pathogen, and they're going to release inflammatory and chemotactic factors. They're going to release vasodilator, the histamine, and heparin. And activate, they're also going to, the mast cells are going to activate basophils. Okay. Now, the basophils on mast cells together are releasing histamine and heparin, and this is going to result in vascular changes. Let me get a little closer so this is a little bigger for you. Okay, so then you have these vascular changes here. Okay, make sure that you're able to list the 3 vascular changes that happen, because this is a pretty important part of the whole inflammation response. And the signs and symptoms of inflammation have to do with this. Okay, so we see, so histamine is a vasodilator, so that would be one of the vascular changes here. So we have vasodilation of arterioles. This is for the blood vessels that have not been injured. Okay, so this is a healthy blood vessel. Because when you scrape your knee, yeah, some vessels get damaged, and the first response of your vessel damage is going to be vascular spasm in those damaged vessels, so you prevent bleeding, right? And then coagulation phase happens and all that. The vessels that are nearby that have not been damaged, they're going to go through vasodilation. So you open up the vessel so that more blood can come in, more immune system cells can be brought in, more proteins that are necessary for repair can be brought in, and basically more supplies. Okay, you're going to increase blood flow to that area by increasing vasodilation. You're also going to see increase in capillary permeability. This is going to help things move out of the capillary faster. More larger things are going to be able to pass through, so we'll see some movement of proteins, as well as it makes it a little easier on the immune cells to move from blood into that tissue. Okay. And also more fluid is being filtered, because we increase the permeability, then fluid gets out easier as well. And then the third thing that we see as far as vascular changes go is display of CAMs. CAMs stand for cell adhesive molecules. So those are those they look like little hooks or something that you see here. Okay, so I'm circling these now. Okay, these are cell adhesive molecules. And what the function of these is that they trap the leukocytes that are in your blood. And so when a leukocyte gets stuck to those cell adhesive molecules, or to one cell adhesive molecule, now the CAM has it, it's grabbing it, it will start bringing it close to the wall of the vessel. This is called margination. Okay, so now we're going to be right here. Okay, so it's called margination. And then eventually, when the cell is close to the wall of the capillary, the cell begins to change its shape, and it squeezes through the spaces between cells of the capillary by the process known as diapedesis. So look at this. Okay, do you see how it's like literally changing its shape, trying to get between those spaces between the cells? Okay, so that's diapedesis. And then the last movement would be chemotaxis. So once that immune cell is in the tissue, it is then going to move towards the source of chemicals. Chemicals meaning those chemicals released by the injured tissue, by these bunch of these bacteria that are in there. Okay, the immune cells are going to be attracted to the chemicals, because they know that wherever these chemicals are coming from, that's where the bacteria and damage is, and so they're going to move towards there. Once these immune cells get to that area, by the way, the most abundant leukocyte is neutrophil. So usually, who we see first at the site of infection are neutrophils. Okay, once these immune cells get to the site of infection, now they're starting to destroy the bacteria and viruses and all the pathogens that entered through that damaged tissue. And we're going to see a lot of dead bacteria and pieces of bacteria that are released into the interstitial fluid. You're also going to see some dead neutrophils and dead immune system cells. And so this fluid, this excess fluid that you have here that contains traces of bacteria and dead immune cells and other things that are a byproduct of basically fighting this infection, this is called exudate. It's basically interstitial fluid with all this stuff in there, and it's an increased amount of interstitial fluid in this area. Because remember how we said capillary permeability increased? Okay, that means that you're going to have more fluid getting into this whole area, and you're going to see some swelling. Okay, so anyway, that fluid from the interstitial space, or exudate, is what is going to enter the lymphatic capillary. Okay, so you can see that here. And then lymphatic capillary will ultimately send it to a lymph node that's closest to the infection. Lymph node, things will happen in the lymph node that we will see when we actually get to the adaptive immunity. But lymph node is going to initiate that adaptive response. Okay, and that is basically inflammation in a nutshell. Some signs of inflammation would be you would see swelling in the area, you would see redness, and increase in temperature, like heat. Well, the area that's going through inflammation is usually warmer. And these things are usually due to, or mostly due to the increased blood flow to the area. Okay, so you see swelling, it's because there's more blood coming to the area and permeability increase, so you have more filtration going on, thus the swelling. The redness, more blood is coming to the area, so it looks red because those vessels are open more. And temperature, more blood again is coming to the area, and remember, blood warms your body, so blood is warmer than your measured body temperature, and so those are the main signs. And pain is also associated with inflammation generally. That's due to some kinins that are in the area that are not mentioned on here, but that's another sign of inflammation. And that's that. Kind of imagine that as this fluid, this extra fluid is being, expelled, or not expelled, but filtered into this area. It is then, taken by the lymphatic system. This serves kind of like as a washing mechanism of the area. We're removing the debris, we're removing the bacteria out of this area and sending it to the lymphatic system. So that's inflammation. And then the last part of the physiological processes, or the second part really, because there's only two that we talk about, inflammation and fever. The second thing is fever. We don't have a picture for fever, but fever is abnormal elevation of body temperature by at least 1 degree Celsius. And fever is beneficial. Okay, fever has benefits. It inhibits pathogen reproduction. So even if you have a low-grade fever, that elevated body temperature prevents the pathogen from replicating at a normal rate. So if you let your body go through fever, of course you don't want it to get too high, okay? So it's important to monitor fever and definitely treat it if it's getting too high. But low-grade fever is good because that elevated body temperature prevents the virus or a bacteria from replicating inside of you. Okay, or they're replicating but just slower, and it gives more chance to your immune system to kill them. The fever also promotes interferon action, so the interferon are being released faster. Remember, interferon warns the healthy cells, and it calls for immune cells to come and kill that infected cell. It also promotes adaptive immunity. Just in general, higher temperature makes your adaptive immunity works more efficiently. And then it also increases formation of CAMs. So your vessels would display, with higher temperatures, CAMs are displayed more frequently. And so you can grab onto more of those immune system cells faster, and you can get them to the tissue better. So those are benefits of your fever. But there are also downfalls. So as I said, you don't want fever to get too high. Okay, it needs to be monitored. It needs to be within a certain limit. And your text, I'm not going to ask you the numbers as far as like when do you get irreversible brain damage and all that. And it actually fluctuates. Those are just average numbers. But if you're interested in seeing that, you can look. I know that sustained fevers of 102 degrees Fahrenheit or higher can lead to seizures in some people. Sustained, and this is sustained. This doesn't mean that you check your fever and it just hit 102 and you're gonna have seizures. Not necessarily. It's sustained. It's if it's for a long time, 100 or above. So the fever, and if your fever gets too high, you can die. Okay. So monitoring fever is very important. But yeah, you don't have to for low-grade fever. If you can manage it, it's better to suffer through it so that your body repairs itself faster. But check it often. Um, every 30 minutes, every hour, if you have to, if you're not medicating the fever, because it could spike pretty quickly. So children tend to have higher body temperature just in general. It's something that I didn't know whenever I had my first daughter. I remember when she first got fever, it got to, I think, 102. And I was thinking, she's a small baby, so I'm like, okay, that's really high fever. She's gonna be in trouble. So I called my doctor, and they're like, okay, you can give her a little bit of Tylenol, and like next time you don't have to unless it's 103 or above. And I'm like, oh, thanks. But now I know. I know people whose kids have really high fever, and that's just normal for them. That would freak me out if my kid had 104. But anyway, so fever has benefits. There is a reason why your body raises its body temperature, but know that it can have dangers also. So you need to be, diligent in monitoring the fever if you are sick. And I'm going to stop there. That concludes our innate immunity. And in the live session, we'll look at adaptive immunity and go from there. All right. See you later.